but not scientific. That's what he's saying about this evolutionary transition between man and human. There just isn't enough there. Second problem. These fossils, the ones that are there, can be widely interpreted. This is one of my favorite stories. National Geographic decided they wanted to see how good all these fossil reconstructions are. Because you know, you never see pictures of the fossils. You always see these drawings of what these fossils supposedly looked at like. So National Geographic said, we want to show people that these reconstructions are reasonable. So they asked 4 artists whose job it is to reconstruct fossils and draw what those fossils were supposed to look like, to reconstruct a female based on one of the skulls the paleontologists believe is a part of the human lineage. One drew a picture that looked like an African-American woman with rather long arms. One drew a thin woman with long arms and a face that looked like a werewolf. One drew a creature whose gender was not apparent. It had a face that looked much like a beaked dinosaur. And one drew a hairy creature with gorilla-like face climbing a tree. The important thing here is all 4 of those artists were completely consistent with the fossils that existed. They were all, and these guys are all trained, these artists were trained to do just this, and they were consistent with what they were given. However, the preconceived notions of the artists ended up determining mostly how that fossil reconstruction looks. And the point here is, if you want to really find out what evidence we have for evolutionary transitions, you don't look at pictures, you don't look at drawings. You look at the fossils. Find pictures of the fossils. You'll be amazed at how little you find. Third problem, there's not even agreement among evolutionists once you get to these fossils. Marvin Lubenow is a creationist college professor, and every year he assigns his students a research project. They are assigned one of the fossils that's supposedly part of the human evolutionary tree. And they come in and go out, and sometimes they're a part of the human lineage and sometimes they're not. But whatever currently is a part of the human lineage, he assigns to his students. He says they have to spend a minimum of 8 hours of research, use only evolutionary sources, and all they have to do is tell him the age of the fossil and where it is in the human line. That's it. Age of the fossil, where it is in the human line. Here's what the students find. They can't report the age category or both because the evolutionary sources disagree with one another. One evolutionary source will put it near the top of the evolutionary line, others will put it near the bottom. The morphology of the fossil, which is what the fossil looks like, is often ignored because of the date. There are some Australian fossils in the human evolutionary lineage, for example, that look a lot like one of the later species that are supposed to be part of the evolutionary line of humans. But they're classified as modern humans because they're dated only a few thousand years old. So even though they look like one of the previous fossils, they are classified as something different because of the way they're dated. And this happens quite a bit. Sometimes morphology is ignored because of date. Sometimes date is ignored because of morphology. It depends on what the source wants. The fossils really can't be put in any kind of evolutionary lineage not based on evolutionary sources because there's no agreement. Now, that's okay that there's no agreement among scientists on any current theory. There's not a lot of agreement on scientists. That's the point. You know, we get scientists arguing with each other and so forth. The problem is these evolutionists who will argue with each other about where to put these fossils in the human evolutionary lineage, when they talk to students, say, oh, we know the human evolutionary lineage. It's very clear. But then when they get together, they say something completely different to each other. That's a problem. As Marvin Lubenow says, it's no accident that evolutionist books seldom include charts listing everything in the human line. One of the more recent texts on human fossils is by University of Chicago professor Richard Klein. In his 524-page work, Klein has 20 different charts dealing with various aspects of the human fossil record. Yet, there is no way a student could get the overall picture. The student would simply have to accept by faith Klein's thesis that fossils demonstrate human evolution. The one chart showing all the relevant fossil material and the lineage is not to be found anywhere. By this type of omission, the true nature of the human fossil record continues to be the best kept secret in modern paleoanthropology. The point is, if we knew that humans really evolved from apes, we ought to be able to show a chart of fossils. We can't. Nobody can agree on them. There aren't very many. And the interpretation of them varies quite considerably. So from a fossil standpoint, we have a hard time demonstrating evolution. To me, one of the most damning evidences against evolution comes from molecular biology, biochemistry. There are certain proteins that exist in most organisms. Now, you've probably heard the term protein before. You know, when you eat steak, you get a lot of protein. What you probably don't need to know is proteins are vitally critical to the chemistry of life. Next to DNA, proteins are probably the most important chemical. In fact, as far as we know, the only thing DNA does is it instructs the cells how to make proteins. That's how important proteins are. Well, there are certain proteins that exist in all or most organisms. All right? Now, proteins are made from smaller molecules called amino acids. And even though the proteins exist in all organisms, the amino acid sequences are slightly different from organism to organism because of different biochemistry. So, for example, one protein that exists in a lot of creatures is called cytochrome-C. All right? And here I've shown a portion, just one section of cytochrome-C for several different organisms. Each one of these three-letter abbreviations represents an amino acid. When you link up the amino acids in just this way, you get a portion of the cytochrome-C that you find in yeast cells. You link them up together in this way, you get a portion of the cytochrome-C that you find in moth cells. So, every one of these guys is an amino acid. The sequence of amino acids determines how the protein behaves. Now, let's look up at the top, look at the horse. Okay? So we've got the cytochrome-C sequence, a small part of it, for the horse. Now, you've got these three-letter abbreviations. Let's compare the horse to that of a kangaroo. Notice that most of the three-letter abbreviations are the same, aren't they? Starts out with GLY, ends with PRO. Most of the intervening ones are the same. However, the one that's in red, ILE, is different. In the horse, the second amino acid is LEU. In the kangaroo, the second amino acid is ILE. All right? Human also has one difference between as compared to the horse. All of the amino acids are the same in this section, except for right here. He's got a THR instead of a THU. So every one of these little red guys here is an amino acid that is different from the horse's amino acid. So every organism has cytochrome-C, but the flavor of cytochrome-C is slightly different from organism to organism. Now remember, DNA tells us how to make these proteins. So in some way, by looking at these proteins, we're actually probing the DNA, because the DNA is what tells it to make this protein. So it would be interesting to compare these proteins over a large range of creatures. Because after all, think about what we're learning. If we look at the differences between the proteins, we're actually looking at the differences between the corresponding sections of DNA. I've got some section in DNA in the horse that's telling the horse, here's how to make cytochrome-C. I've got another section telling the kangaroo how to make cytochrome-C, and it's just slightly different. By comparing these proteins, what we're really doing is comparing those sections of DNA between the horse and the kangaroo, the human and the chicken. Now, think about what we should assume if evolution were true. From an evolutionary point of view, cytochrome-C exists in every creature because it was inherited from a common ancestor. We start off with a very simple common ancestor, and as time goes on, things get more and more complex. If I look at the creatures I had before, yeast, wheat, moth, tuna, pigeon, horse, okay? I can order them this way in terms of increasing complexity. A yeast is a single-cell organism. Wheat's a plant, moth's an insect, tuna's a fish, pigeon's a bird, horse is a mammal. This is an increase in complexity. Now think about what evolution says. Evolution says all of these guys originally started off as some very, very simple life form. And as time goes on, it became evolving. And at first it produced the lines that produced a yeast, because the yeast are pretty simple. So we start off with a very simple life form. The first kind of life form it's going to evolve into has to be simple as well. So early on in the evolutionary passage, the yeast formed first. Then most likely later on the wheat formed, then the moth, then the tuna, then the pigeon, then the horse. That's the evolutionary framework. Now if that's the case, if I took a very simple life form and I compared the cytochrome-C of that simple life form to that of a yeast, I would expect them to be pretty similar. And if I compared that simple life form to that of a horse, I would expect a lot of difference. Because from an evolutionary standpoint, the yeast came a lot earlier. Not nearly as much time for mutations and all that to occur. So in the end, if I were to take a bacterium and look at its cytochrome-C and compare how